Hi, this is Alex from devstories.eu. AlphaFold Protein Structure Prediction, Episode 13 of 21. DeepMind open-sourced their latest structural biology model, but if you clone the repository and try to run it, it will fail immediately. The code is entirely public, but the actual neural network driving the predictions is tightly controlled. Today, we are looking at the AlphaFold 3 inference pipeline and how you actually navigate deploying it. To run AlphaFold 3 locally, you must unite two separate pieces, the execution code and the model parameters. The repository gives you the pipeline logic. For the parameters, specifically the version 3.0.1 weights, you have to request access separately. Google requires you to submit a form detailing your non-commercial research use case. Once they approve your request, you receive a link to download the heavy model weights. This split approach allows DeepMind to restrict commercial use while still letting researchers inspect and run the open-source pipeline on their own hardware. After downloading the weights, your next step is defining what you want the model to predict. Earlier versions of AlphaFold mostly dealt with single protein chains, so you could just pass a simple text string of amino acids. AlphaFold 3 models complex interactions across diverse molecule types, so a basic string is no longer enough. Instead, you create a configuration file. This file acts as a complete manifest for your biological target. You specify the exact molecules involved in your experiment. This might be a standard protein, a strand of DNA or RNA, or a specific small molecule ligand. In this same configuration file, you also define randomness seeds, because the new model architecture relies on a diffusion process to generate the final three-dimensional structure. Introducing slight variations in the random seed produces different possible structural states. By defining multiple seeds in your configuration, you instruct the pipeline to generate distinct predictions for the exact same input. You also declare a dialect version in this file, which simply tells the parsing engine to apply the AlphaFold 3 validation rules to your manifest. With your weights downloaded and your configuration file ready, you move to execution. Because this pipeline depends on highly specific versions of machine learning libraries and GPU drivers, running it directly on your host operating system is risky. The official workflow relies on Docker. You build a container image directly from the provided repository, locking in the perfect environment. To actually run the inference, you launch this container and connect it to your host machine by mounting three critical paths. First, you mount the folder containing your input configuration file. Second, you mount the directory holding those massive approved model weights you downloaded earlier. Finally, you mount an empty directory where the container can write its results. Once the container boots, you invoke the main run script. The pipeline validates your input manifest, loads the weights from your mounted directory into GPU memory, and begins the structural prediction. When the process finishes, your mounted output folder will contain the final coordinates of your requested molecules, formatted and ready for visualization. The primary challenge of this pipeline is not the underlying code, but managing the logistics of securing the model weights and meticulously structuring your configuration file to map out complex biological interactions. If you would like to support the show, you can search for DevStories EU on Patreon. That is all for this one. Thanks for listening, and keep building.